Nephrons regulate the composition of blood by a combination of three processes that transfer materials between the nephron tubules and the capillaries that serve them: filtration, secretion, and reabsorption. A nephron tubule and the blood vessels surrounding it have been straightened and simplified in this diagram. The gradation of color, lighter on top, darker on the bottom, corresponds to an increasing concentration of solutes in the interstitial fluid surrounding the nephron toward the center of the kidney. As blood flows through the glomerulus, a knot of porous capillaries, water and virtually all other molecules small enough are forced by blood pressure out of the glomerulus and into Bowman's capsule. This process is called filtration, and the fluid that accumulates in Bowman's capsule is called filtrate. Blood contains blood cells, plasma proteins, water, sodium chloride, hydrogen ions, bicarbonate ions, urea, glucose, amino acids, and possibly some drugs and poisons. Blood cells and plasma proteins stay in the blood. They are just too big, but all the other substances are filtered from the blood and form the filtrate that accumulates in the nephron tubule. As the filtrate moves along the proximal tubule, certain substances are transported from the blood and added to the filtrate, a process called secretion. Secretion of hydrogen ions helps to regulate body pH. Certain poisons are also secreted to remove them from the blood. This list summarizes substances contained in the filtrate. Filtration is not selective, so some of these substances are useful to the body, and it is important that they be returned to the blood. The kidneys generally need to recover water, sodium chloride, and nutrients such as glucose and amino acids. This reclamation of valuable solutes in water is called reabsorption. Let's look at the process of reabsorption as the filtrate continues its journey through the nephron tubule. In the proximal tubule, reabsorption of bicarbonate ions helps regulate the blood's pH. Sodium chloride, glucose, and amino acids are actively transported out of the filtrate. This leaves the filtrate more dilute than the surrounding interstitial fluid, so water follows by osmosis. These solutes and water re-enter the blood.